You watched the video I showed you? Anybody watch any other videos? Alright, good. Cell division, mitosis. 4 general phases. Remember this, write this down, put a star beside it. Mitosis is formation of 2 nuclei. You're replicating the genetic material. You're replicating the genetic material. You're making 2 nuclei where there was only 1 before. What's the function of the nucleus? House of DNA, yes. Nucleus house of DNA. So, mitosis is nuclear replication. Mitosis makes 2 nuclei, not 2 cells. Big difference. Cytokinesis makes 2 cells. Mitosis makes 2 nuclei. Big difference. You should know the difference. Haven't made enough noise about that yet. Cytokinesis is division of cytoplasm. You end up with 2 cells. What's the name of the structure that forms during cytokinesis? Cleavage furrow, yes, that's a cleavage furrow. That's where the cells pinch off. Do you need to watch another cell cycle video? Or are you okay?
We're okay.
You're okay?
Yes.
Alright. So, interphase, mitosis, blah, blah, blah. Protein synthesis, on to some new stuff. Protein synthesis is making protein. For protein synthesis, we use RNA. RNA is chemically very similar to DNA. Chemically very similar to DNA. There's 3 different kinds of RNA used to make proteins. mRNA stands for messenger RNA. mRNA stands for messenger RNA. You could say it carries the message. mRNA is the actual copy of the DNA that codes for the protein. mRNA is the actual copy of the DNA, codes for a protein. DNA is very precious. You don't want it floating around in the cytoplasm. It stays in the nucleus. Messenger RNA is copied in the nucleus and sent out through nuclear pores into the cytoplasm. That literally is the code or the recipe for a protein. How are we doing, everybody? Okay? rRNA stands for ribosomal RNA. rRNA stands for ribosomal RNA. Ribosomal RNA is what ribosomes are made of. Ribosomal RNA is what ribosomes are made of. What do ribosomes do? Make proteins. Ribosomes make proteins. They're like little machines that assemble the proteins amino acid by amino acid. Take the floor of a factory, it's going putting stuff together, putting amino acids together. And it uses the messenger RNA to determine what amino acid comes next. It uses the messenger RNA to determine which amino acid comes next. I'm going to show you a video of this in a minute. Okay, we've been talking about ribosomes. What makes ribosomes? Nucleoli found inside the nucleus. Yeah, good. The third kind of RNA is called tRNA. That's also known as transfer RNA, or that stands for transfer RNA. Transfer RNA brings the raw material. transfer RNA brings the raw material. In other words, it brings the amino acids. Each piece of transfer RNA has one amino acid hooked to it. It's going to bring it to the ribosome and say, here, this is next in line. It gets added to the growing chain. When I think of this, I think of the baby toys. I don't know if they still make them or not, but they were like these plastic little shapes that had two ends. You could make them together into a big long chain, or make them into a necklace, or like a circle and a tube and a triangle and stuff. You know, stick them all together. That's what I think of proteins is like, with all the different amino acids. And this whole setup, the messenger RNA says what amino acid comes next. The ribosomal RNA is where you make it. The transfer RNA brings the next block and plugs it in. Probably kind of like to see a video, right?
Mm-hmm.
So this is not the one that I normally use. I just did a quick search and this one pops up at 2 minutes and 42 seconds long. You want to take a chance on this one?
Yes.
Sure, let's do it.
Which one is it?
Not like you had a choice, we're going to do it anyway. I like you guys to think you have some control.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes.
Okay, that's a chromosome there, right?
Mm-hmm.
If you uncondensed it, what would you call it? Chromatin, yes.
Each chromosome contains a long strand of DNA, tightly packaged around
What are those proteins called? Histones, good.
proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus.
See how this guy's matching up with this guy's matching up with this guy. A always goes with T, U always goes with A, G always goes with C, C always goes with G. See how that's doing? It's going to make kind of like a mirror image copy. This is the messenger RNA. And this process is called transcription. I want you to write this down. Transcription is the process of making a messenger RNA copy from DNA. Transcription is the process of making a messenger RNA copy from DNA. That's transcription. This is taking place in the nucleus because the DNA is there. Everybody okay?
Are we going to need to know the A is T part data?
I'm not going to ask you which base pairs match up, but as a science student, you should probably know. G always goes with C. A always goes with T. You know, T is on one side, A is on the other side. Now this is for DNA. And I remember this because if you spell it out, G-C-A-T, G, it's a cat. Okay? G-C-A-T, G always goes with C, A always goes with T. Now in RNA, it's a little different. RNA has G's and C's. It also has A's, but it doesn't use T, it uses the uracil, which is a close chemical relation. So A always goes with U. DNA and RNA. Again, I'm not gonna ask you this on a test or quiz, but it's good to know, just in a general sciencey thing. And if you want some even more sciencey stuff, every protein starts with TAC, which translates into AUG, that's the start codon. That means start making a protein here. You can remember that because school starts in August. Again, you don't need this, but if you want to remember it, if you're taking more classes, more bio classes than this, it'll help you. Maybe even microbiology.
The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA.
Here's our rRNA that's making a ribosome. There's our mRNA.
The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids.
And that's the tRNA, or transfer RNA. There's the amino acid. This thing right here is called the anticodon, and we'll see what it does in just a second.
There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time.
See how they line up? These 3 bases match up with the opposite 3 bases in the messenger RNA, and that's how you know you're bringing the proper amino acid. Because these 3 guys are coded to the amino acid it's hanging on to. This one will only bring that particular amino acid. That's how it makes sure that everything's right. This is a codon, and this is the anticodon.
As each triplet is read, a transfer RNA delivers the corresponding amino acid.
Now,
This is added to a growing chain of amino acids.
by the way, this process is called translation. Translation is actually producing a protein. Translation is producing a protein. If you want to be really fancy, you can say translation is using mRNA, rRNA, and tRNA to produce the protein. It's using all 3 of those different RNAs to make a protein. Transcription is making the mRNA copy. Translation is using all 3 RNAs to make a protein.
Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
What stage of protein structure is this? Pardon? Tertiary. Because it's all folded as one of them, but it's all folded up. Back here, what stage would that be? Primary. If you had a couple springy things in, it would be secondary. How do we feel, okay? That's transcription and translation. Are you guys okay with that? Question.
So the reason why they make it mirror inside the nucleus is because the transfer protein actually makes the right one the opposite of that.
Correct.
Mirror. Okay, cool.
But the transfer RNA, remember, is RNA, so it's using uracil instead of thymine.
Well, yeah, same. So that's
Yeah.
concept that's kind of dog.
Other questions? Yes.
What is the U called?
Use called uracil. So we're all okay with that chapter? All right. One more to go. And you guys should be deep into this in lab, right? I know my guys are. If you don't have me for lab, you're already doing tissues, right? All right, good. So, what's a tissue? Definition. You know this. The more cells working together for a particular function. There are more cells working together for a common goal or particular. And what's an organ? There are more tissues working together. Is a bone an organ? Yeah. Is a nerve an organ?
Yeah.
Yeah, nerve cells and supporting cells. Is a blood vessel an organ? Yeah. Alright. Histology is the study of tissues. Histology is the study of tissues. Most of our tissues, when you slice them thin enough to see through them, they're clear. So we have to stain them. You guys have been looking at slides, they're all stained. They're all stained, that's what makes them easy to see. Four basic types of tissues and their general roles. In general, epithelial tissues cover surfaces and line cavities. Epithelial tissues cover surfaces and line cavities. Your skin is an epithelial tissue on the outside. In general, connective tissues provide support for the body and connect things together. In general, muscle tissue provides movement. Whether it's moving your body, or it's moving things inside your body like blood or lunch. And then in general, nervous tissue controls many different functions in your body. Nervous tissue. Another way, a better one is nervous tissue sends messages. Nervous tissue sends messages. So those are gross overviews. We're going to get into much more detail over today and some of Wednesday. Epithelial tissues, two major types: epithelial tissues that cover things or line things, and glandular epithelium. Covering epithelium forms the surface of your skin. It also goes down into the cavities of your body. Every place that's not attached to another tissue, that's an epithelial tissue. Glandular epithelium is found in glands. They produce hormones and digestive juices and things like that. Functions of epithelial tissue. Epithelial tissue protects underlying structures. Epithelial tissues protect underlying structures. Epithelial tissues absorb different chemicals. They absorb nutrients. They absorb water. Epithelial tissues filter your blood. Epithelial tissues filter your blood like in your kidneys. Epithelial tissues excrete waste. Epithelial tissues excrete waste. They also secrete products. They also house sensory receptors. Everybody okay? Any questions? All right, characteristics of epithelial tissue. Epithelial tissue is pretty much all cells. We say it is cellular, or it has cellularity. Epithelial tissue is almost all cells. There's not much in between the cells. Those cells have junctions in between them. The cells have junctions in between them to help hold it together. Especially tight junctions and desmosomes. You guys remember what tight junctions do? Nice, zipper-like structure that forms a waterproof barrier. Zipper-like structure that forms a waterproof barrier. What about desmosomes? What do?
They're the anchor point.
Yeah, anchor points, really strong connections between cells. You don't want to get your cells torn apart. Epithelial tissues have polarity. Means they have two sides. The apical surface is also called the free surface. The apical surface is also called the free surface. That means it's not attached to anything. On the apical surface, you will find microvilli, some places. Other places you might find cilia. What are microvilli? Now, what they do, what are they? Yeah, think of like extensions of the plasma membrane. What do? Increase surface area. Yeah. What are cilia? Yeah, hair-like structures, and what the cilia do? Move material, move mucus. If you did not know the difference between those two, you got a lot of work to do for Monday. basal surface is the bottom of the epithelial tissue. Basal surface is the bottom of the epithelial tissue. It's connected to the underlying vector tissue. It's connected to the underlying connective tissue. The basement membrane is the structure connects the epithelial tissue to the connective tissue. Basement membrane connects the epithelial tissue to the connective tissue. What epithelial tissue am I in the middle of drawing right now?
Simple columnar.
Yeah, simple columnar epithelium. A simple columnar epithelium, you should know that from last week.